Well, first, thank you for having me here. It's a thrill to have a chance to share the work that I and the much larger dog genome community and dog genetics community have been doing for the last several years. So, being able to understand the architecture of the genome and eventually the actual sequence of the genome of many kinds of canids, including many, many breeds of dogs, has really opened up our thinking about the timing of dog domestication from wolves, the locations where that happened, the context of human interaction with dogs during that domestication process, what sorts of modifications the genome had to undertake in order for dogs to survive, and then in order to enhance human survival. So, for instance, we know that the dog's diet changed. And so we know that there are changes in some of the enzymes that are really important in processing starch. And that's something that had to change in order for dog survival. But we also know that as dogs became more and more and more important for human survival, as they aided in hunting and they aided in guarding and they aided in herding, that there are behavioral changes as well. And those really interplay as we learn about human migration and human survival. We learn more about dogs. As we learn more about dogs, we learn more about humans. And those things really play off one another. So we would love to understand what are the genes or the variants or the different alleles, as we might say, that underlie the different behavior patterns that you see across dogs. And so we tend to think of herding dogs or dogs that assist in hunting or pointing dogs. We think of those as sort of siloed behaviors, but they're really not. There's no one behavior that's owned by any one particular dog breed. But that doesn't mean there isn't a lot of underlying genetics. And lots of labs are looking at that right now, in part because we think it'll inform our studies of human health and human biology and human development. And so people often draw the analogy of herding dogs. They're very smart. They learn very quickly. They execute repetitive behaviors. They can take signals very well from an owner or herder. They're very goal oriented. And so understanding that underlying genetics, what happens when we look at those genes in humans? What happens when there are different variants in the DNA sequence in humans? Is it going to reveal something about human behavior, something about normal human behavior, something about maybe a more neurodivergent human behavior? We're constantly thinking about one versus the other, this sort of one health approach. It's not really the right approach to think about dogs over here and humans over here. Dogs have come up through with humans, right? They've I don't want to say evolved, right, necessarily, but they're integral to our survival. And so we want to look at them together. Yeah, well, dogs live in our environment. They eat our food, they breathe our air, they're exposed to, our pesticides, our pollutants, all the good and the bad. And so, much as our health reflects our environment, their health reflects our environment as well. And it's probably not surprising to know that dogs get the same diseases as humans do for the most part. And yes, there's a big emphasis on cancer, but you can think of a variety of diseases. Think of epilepsy, think of diabetes, think of motor neuron diseases, think of osteoarthritis. I mean, you name it, and there's probably a dog disorder that's reminiscent or similar or recapitulates key features of that human disorder. So the advantage we have in studying dogs though is breed structure, right? And so if I'm looking at a particular disease in boxer, I can look at the same disease in breeds that are related to the boxer, whether it's the bulldog or the bull terrier or the dog de Bordeaux, right? And I can hypothesize that whatever mutation is responsible for the disease in the boxer, it's probably going to be the same in the other breeds because they share recent common ancestry. And it's really hard to do that kind of study in humans because dog breeds come with labels. I'm this breed. There are 350 dog breeds. This, and this dog breed are, Scottish Terriers, and and this dog are Border Collies, and and this dog are Golden Retrievers. And so that offers tremendous advantages for reducing all the statistical noise and complexity that we get when we study diseases in humans. So finding these genes in dogs, which is what we're now doing, and then looking at those same genes in humans is helping us identify parts of genes that are really critical for function or for execution or for structure of the encoded protein. And we certainly look at mutations that have been identified in humans associated with particular deleterious or diseases, and we look at those in dogs. And again, we put those things together. So I think it's going to be this ever growing sort of exploding process as we think about more and more and more diseases that we want to understand in both dogs and in humans. Yeah. Um, So I don't know that our overarching goals changed a lot. We wanted to understand the genetics of behavior in an organism that had pretty sophisticated behavior patterns. We wanted to see how population structure could enhance those sorts of studies, which is what you get when you get 350 breeds. We wanted to find disease genes. We knew that would help understand human health and human biology. We wanted to understand the impact of environment. If you live in a household where there is smoking, does secondhand smoke up the chances you're going to get cancer if you're a dog? Right? Those are really fundamental questions. Um, and all the other sort of obvious things about behavior and morphologic variation has been a big interest of my lab since the 1990s. The biggest to the smallest dogs differ in size by 40-fold. How do you get that within a single species? And in such a short period of time, right? Dogs were domesticated 20, 30,000 years ago, and most breeds have only been around for a couple hundred years, were developed in Europe during Victorian times. How do you do that? What are the genes that have to be hit, and how do they have to be hit to do that? So I think many of those questions you could put under the broad label of variation. Dogs have tremendous variation, morphology, disease, behavior. How do you find it? So I think those have always been there. I think what's changed is what we know about the genome. So the first sequence of the dog genome in 2005 jump-started the field just remarkably. It gave us so many tools and so much insight. Um, and that's just been expanding. I mean, the community recently published its first iteration of Dog 10K, which is 2,000 canine genomes across, a huge number of breeds, as well as village dogs and non-breed dogs, mixed breed dogs. And so that's given us a huge resource to understand, well, what are the parts of the genome where there are repetitive elements? What are the parts of the genome that are rearrangements? What are the parts of the genome that are constrained, that are common to all dogs, that are just invariant, that you really can't seem to mutate? And where is the greatest amount of variation? And the more breeds we have sequence from, and we know what those breeds look like, because to be a member of a breed, to be a registered member of breed, your parents had to be as well, and your grandparents and your great grandparents, right? So as we match up all those sequences with all of that variation, I think our world is just, exploding. We have endless opportunities to study anything that we want to. And I think as people have really mastered studies of ancient DNA, I think that's just added to the fabric, right? So ancient DNA, dog bones all over the world, what we know about those particular sites in terms of humans and what we find in terms of dogs and the position of the bones and what the humans were doing or what we know about that human culture. That's just really added to the fabric. But I think it just fundamentally comes down to variation. So someone can come into my lab and I can say, what do you want to study? And they'll say, well, do you have a project on? I'll say, Almost certainly, because we study variation. And which direction do you want to go? Which flavor of variation do you want to look at? And it's just extraordinary. It's extraordinary what's been captured in the dog genome and across dog breeds. So thank you for that question. There's probably two or three things. My background was in sort of DNA structure, transcription. And after I finished my first postdoc, I knew I really wanted to do something different. And I wanted to work within a biological system. So the idea of studying dogs had been bubbling around because, everybody looks and recognizes a breed and has an opinion as to what they're going to be like, their personality, their behavior, their traits, right? But there were two papers that were published simultaneously, back to back. And both papers described a kind of a marker in mammalian genomes that would be great for making maps to navigate around the genome called microsatellites. And they were great because they had enough variation that there was the ability to study a population. There was variation across a population. But they were stable enough that you could look at the inheritance of the surrounding genome through generations of a family. So they sort of hit that sweet spot. And that meant you could make a map of any mammalian genome. You didn't have to be a big, huge, fancy lab to do it. You could study the biology of any mammal you wanted to study, and it was very doable. It wasn't some great, big, huge pie in the sky kind of project. And so I was very fortunate to be in a lab at Berkeley where I had that opportunity and started the development of the canine genome map, where the lab was really supportive of that idea, and then just continued on. I always thought, well, I'm not sure how long this dog thing will continue. I may have to switch to studying this or that human disease or trait. But 35 years later, I'm still just studying dogs and having a great time doing it. And I will say, over those years, the community grew and grew and grew, and just fantastic people came into the community with very original ideas, very creative ideas, very innovative people. And the human genetics community kind of got us. They're like, yeah, you have a really important place in understanding human health and biology, and welcome. And so all those things made it so much easier and so much more fun. So one thing I want to add is that integral to the success of all of our labs has been this concept of citizen science. And so we don't breed dogs, we don't kennel dogs, we don't tell people how to breed dogs. We work with pet owners like everybody out there, and collect samples from their dogs, collect medical information or American Kennel Club registration information or mixed breed dog information, whatever information they have to share with us, pictures of their dogs, stories of their dogs, measurements of their dogs, but whatever we can get. And we've built our entire research program around those 40,000 DNA samples. And I can tell you, researchers around the country who study dogs are doing the same sorts of citizen science projects where we want to know about the dogs that are really out there, that are living in your homes, that are part of your family. How do they interact with your family? How does your family interact with them? And then what's special about that breed? What behaviors, what morphologic traits? And the only way to really do that is to invite the general public to, hey, be part of our science, be part of what we're doing here at the National Institutes of Health. And that has been the most rewarding and successful approach to studying dogs that one can imagine. It's been terrific. So you can go to our Facebook page, and it describes a lot of our studies, and it actually has a list of dog breeds that we don't have enough representation from. And if you have a dog of one of those breeds, for sure, contact us through our Facebook page. We'd love to get a DNA sample and get your dog enrolled in the NIH NHGRI Dog Genome Project. And if your dog has a particular cancer, there's cancers we're focused on, like bladder cancer, or histiocytic sarcoma, and in particular breeds. And so if your dog fits into those profiles, for sure we want to hear from you. You can also just look at the National Institutes of Health websites. And we're in the National Human Genome Research Institute. And you can look there and you'll find more than you ever wanted to know about the Ostrander lab, probably, and all of the things we do. And there's other dog genetics labs in the country that are focused on other health issues that are just fantastic. And if we're not studying whatever it is your family's interested in, your dog needs to have attention to, we can almost certainly point you in some other direction as well.